Welcome to this episode of Let's Talk Medicine, and we're talking about a very interesting topic today, and that's phage therapy. It's really a paradigm shift in the treatment of infections, and I'm proud to say that Australia is leading the way in this therapy at the moment, and our special guest is Professor Ruby Lin at the Westmead Hospital and at Sydney University. Welcome, Professor Lin.
Thanks for the opportunity to talk about Phage, Nello.
Yes, no, that's a pleasure. Now, before we maybe get a little introduction into yourself and how you came into this, I'd be just interested for the audience to just get a quick definition as to what a phage is or a bacteriophage is. So could you just talk us through what that is?
Yeah, sure. I am a professor in genomics. This whole operation is actually led by Professor John Iredale. So him and we've been working for almost 8 years now, and we actually co-founded Phage Australia. So it's a network of clinicians and researchers and industry. So what is a phage? Well, phage is a virus that attacks only bacteria. So as you're sitting there, you probably have 3 to 5 billion of phages on you and in you. It specifically target bacteria, so it becomes very specific. And what it does, it lands on the bacterium and it actually injects its DNA content and use the bacteria's genetic material and amplifies it itself. So it explodes, if you can imagine something invading and make the bacterium explode. So that's how they take over. So it amplifies it itself as well. So it can replicate, and that in medicine, it's almost like auto-dosing. So it makes it a very powerful tool in medicine.
Fantastic. And how did you come to become interested in this therapy? And can you tell us maybe a little bit of your background, how you became a professor, what your initial research was in, and how you led to leading the Phage project?
Wow, how long have you got? Actually, I
As long
came
as you want,
across
keep it digestible.
I came across phage therapy by serendipity, actually. So my training is actually in cardiovascular diseases, systems biology, transcriptome therapeutics. So dealing with eukaryotic stuff. So I was looking at inherited diseases of hypertension as well as coronary artery disease and looking for gene signatures and doing a lot of the buzzword now, machine learning, look for predictive factors. And then I was doing biobanking and a bit of cancer biology, and this job came up to run a clinical trial. So I was recruited by John Iredale because of my skill set. Yeah, so came in and I realized that I didn't want my children to grow up in a world where antibiotics doesn't work. And, the more I dig into antimicrobial resistant or antibiotic resistance, it's just such a horrendous picture. If you can imagine that you have an accident and you go to the hospital and antibiotics doesn't work. It's just, it's a nightmare,
It's
right?
terrifying. It is
Yeah,
terrifying.
exactly. So that's how I dived into it. And the community is a very small niche community. And I didn't realize, with the human genome and with all the data sets, it's all based on human rat models, mice models and things like that. So I actually didn't really dive into microbiology until I joined this group. And I actually now I can appreciate the diversity of bacteria and the diversity of phages.
Fantastic, fantastic. So one of the things I found curious about Phages is that in some ways it's cutting edge, leading the way to this new paradigm of treating infections, but in other respects it's been around for actually quite a while,
Mm.
particularly in places like Georgia, Russia, those sorts of countries. So can you explain to us a little bit the history of it and why in some respects it's so new and in other respects it's so old?
Yeah, sure. Phage therapy has been around for a century. Several cultures have actually used phage without realizing it's phage for antibacterial or antiseptics. So phage research actually began in the early 20th century. And scientists have discovered that, okay, certain viruses seem to kill bacteria only and doesn't seem to affect humans. So the first documented one is in the 1920s by Felix Harel, and he treated patients with dysentery with Phages. So, and there seems to be, a few, publications, anecdotal stories. But then, in the 1940s, when Alexander Fleming, well, in 1920s, and now during the war, antibiotics become the norm. People seem to have resumed and used the Western, medicine, and went with antibiotics. But there's an interesting twist. So, people don't want to talk about it. There's actually a geopolitical twist to this. Stalin actually played a significant role in promoting phage therapy in the Soviet Union. So if you actually dig into the history of phage research and phage therapy, you will find that a lot of the literature are in Russian or in fact in Georgian. So George Iliava was a Georgian microbiologist, and he's one of the pioneers for phage therapy, and he collaborated with Felix Harel, who discovered phage. So he actually founded the Iliava Institute for phage therapy in Tbilisi, Georgia, back in the 20s, and it's become a center for phage research and phage therapy. So it's been around for ages. And I think it's because of antibiotics use and how we view Western medicine. It's kind of lost its popularity, if you want to put it. And of course, antibiotics is easier to produce, and it's easier to, let's say, it's more commercializable. And so people tend to use it. And it's now people realize that, bacteria co-evolve, and they evolve very quickly. And so now we have this antimicrobial-resistant crisis, and now people are actually going back to phage research and realize that you can actually use a lot of modern technologies to study it and make it very precise. So I think the beauty of it, the main point of why we are coming back to phage research and phage therapy is because a specific phage will target only a specific bacteria. So it makes it, so if you put, the modern terms, precision medicine on it, it's very precise. And because now we've got genomic technology to sequence it, to be able to identify specific sequence gene signatures that's associated with targeting the specific bacteria, as well as targeting the specific, bacterium surface receptor, it makes a very powerful tool.
Now,
Yeah.
with genomics and modern technology,
Yeah.
it's almost like a rebooting of the whole project, in that we can be a lot more targeted and a lot more scientific about how we...
Yeah, so there are a lot of randomized clinical trial now that test the veracity of, so for example, we call it phage cocktail. So you can put a number of phages in one phage product to treat patients. And a lot of those, they put everything in a cocktail together, say 4 phages. It is because that combination would target a whole population of bacteria. So it's not just will kill one clade of bacterium. It's like the whole, so you would need to do genomic epidemiology of the bacterial infection in order to understand and then select the phage specific target that. So you probably, if people read phage therapy, they will probably go, oh yeah, if selected phage that would kill, say, 90% or 99% of the bacterial population.
Okay, so a few clinical general questions just to help us understand this better. The phages are aimed at bacteria. Are these viruses also able to attack other things like fungi or other types of microorganisms, or is it only bacteria that they attack?
The ones that we use attacks bacteria. I'm sure there are other viruses that attack fungi, but I'm not in that field, so I can't actually dive into that.
Now, with the bacteriophages that you're researching and now taking towards clinical trials and taking to market, is resistance a possibility? Can the bacterium become resistant to it? And can the virus itself, the phage, mutate and cause problems of its own?
Yes. So actually, I was in a talk for Global Phage Therapy Clinical Round last night. The Belgians actually presented their experience. They had, and it's published. You can look it up. They've published recently 100 consecutive cases that they have used phage to treat these hard to treat infections. And what they've identified is that, they do co-evolve. So bacteria and phage, they do co-evolve. So you actually do need to keep an eye out for developing resistance. So it's either the bacteria developing anti-phage antibodies, so that's the bacteria. But you're looking at three different interactions. So you're looking at the interaction of phage with the bacteria in the patient. And so from my perspective, when I look at the patients, so human transcriptome, their gene signatures and their immune response in the presence of bacteria and phage, you do develop resistance. With the cases that we have seen so far, and the data that we've got, it's very low. And if you do, then, phage doesn't work, right? So of course you pick another phage that will attack that evolved bacterium or the infection. The beauty of it is that with the resistance, so for example, the discussion yesterday was actually about picking a cocktail of phages that attack different surface receptor of the bacterium that's causing the infection, and therefore minimizing the resistance. So there are actually a lot of research going in at the moment to look at this area. So it is still, quite a bit of R&D to go.
Can you use phages at the same time as using antibiotics, or must they be kept separately?
At the moment, a lot of the cases is adjunct to antibiotics. And I think it's, in Australia, if you do get admitted to the hospital, you do have to administer antibiotics, right? So with our protocol, so from Phage Australia, John Iredale devised this protocol which we have implemented, and it is to standardize therapeutic monitoring and also standardize, phage therapy. And from that perspective, we do look at how the phage behave.
Yeah, no, that's good. So for those of us who haven't had a lot to do with it yet, these therapies, do you give them orally? Do you give them intravenously? How do you administer them to patients?
In Australia, according to stamp protocol, actually a lot of cases are intravenously administered. So we have special permission from TGA, and also an exemption to administer the phage intravenously. The beauty of phage therapy is that you can have different formulation. So, for example, a lot of patients that we have with lung infections, cystic fibrosis patients, etc., the clinicians have treated them with nebulized phage. Quite a few patients with, for example, prosthetic joint infections and things like that, we have actually treated them with a wash, with a phage, open wound wash, or it can be topical. So we actually have formulated phage to just as an ointment on open wounds.
So will there ever be a day where, for example, you go to your GP, you have an infection, and instead of getting an antibiotic prescription, you get a phage prescription? Or is a phage treatment a lot more individualized and won't lend itself to such a sort of broad approach?
Well, I think to avoid antimicrobial resistance, I think that's the ultimate dream, right? I mean, it's going back to the question you asked, at the moment, with all the therapy in Australia, it's adjunctive. So the patient are already on antibiotics and you give them phage. Whereas, a lot of randomized clinical trial now, phase 2, they would actually have an arm. In fact, we're about to start an arm of clinical trial using Staph aureus phages, and it would be randomized to one group of patient doesn't receive phage therapy and the other group, without antibiotics does receive phage therapy. It's really hard because these patients, once you get to that stage, they are already very sick. So you can't actually withdraw antibiotics, right? But the first clinical trial we did using a company's phage cocktail, we actually have identified that it is safe and it is feasible to actually have phage administered alongside antibiotics in these very sick patients.
So are the phages stable enough to be putting into tablets and capsules and storing on a shelf, or are they things that you have to prepare and use straight away?
Oh, that's a billion-dollar question, right? So a lot of companies are trying to look at stability of phages, right? And I think, when I was in Iliava in Tbilisi, I think a lot of scientists pointed out to me, how look, these vials have been sitting there for over 30 years and they're still, stable. So it depends on the phage itself. And of course, companies will have trade secrets and things like that about the stability. But in general, we found, from our experience, if it's purified phage, it would stay stable for at least, 18 months to 2 years. But don't quote me on it because the behavior of individual phages is very different. So it's going back to your question about individualized treatment. If it's a outbreak, of course, it's the same bacterium that's causing the issue, then of course you can massively produce the phage to treat that whole environment. But if it's individual, like a lot of cases that we see now, because we're up to 53 cases, we have seen patients needing the same phage that we have produced, the same batch over and over again, for example. So then the formulation would be, the same, the stability of the phage would be the same. So it becomes a biomanufacturing kind of question. In terms of formulating it, for example, in spray or in different applications, there are companies now. So for example, there is a company make Listeria phage as a spray. So you can actually spray on commercial kitchen utensils and, work surface, for example. There's a company in Poland that actually have formulated phage into animal feed and they give it to poultry in that industry. So there are several applications. At the moment, we just don't have enough data to look into PKPD, so the kinetics of how the phage actually behave, say a chicken or in a cow or in the patient.
So I guess obviously using it for an industrial surface is very different from putting it into a live organism that where it can
Yeah, so
reproduce.
exactly. So the bioavailability, so it becomes a pharmacokinetic question, but even before that, the purity of it would be in question as well. So for example, without biomanufacturing, it's a regulatory debate, or it's a regulatory question that's being debated at the moment from different jurisdictions. So FDA have got their own rules, and it looks at toxin levels, and EMA have got their own rules, and TGA have their own rules as well. I mean, of course, we all talk to each other, but in preparation for human application, for intravenous application, of course, you need it to be at a specific standards. So, for example, the endotoxin for intravenous application would have to be less than 5 EU per kilo, right? But if it's in the feed or you drink it as a probiotic, for example, for oral, application, then the purity doesn't have to be to that standard. And so there's a lot of debate about how you actually manufacture a phage for these different applications.
Fantastic. So given that we're at this stage now where there are clinical trials and there's some patients are being successfully treated, where do you see the role for doctors in the community getting involved or learning more? Do you have any suggestions for what we should be doing as a medical profession now?
Well, I think educating the doctors, and of course the pharmacists, the nurses, we need to have the buy-in from the community, from the consumer, so from the patient community itself, as well as from the medical community. So, for example, when we first ran the clinical trial, the nurses were really worried about phage. They don't know what it is. And as soon as you say it's a virus, you automatically have this fear. When we always talk about phage needs a new branding, because if you mention virus, then, people stand back. We do put out, part of the Phage Australia, we do put out education materials to inform people what phage therapy is, and it is safe. I think the community now do recognize that it is safe to use and it is feasible to use, but now we just need to have this regulatory framework around it to make sure that we're using it properly. In terms of adoption, I think with cystic fibrosis community, so we did a survey with CFA, Cystic Fibrosis Australia. They are the first adopters of all the new technologies, because they're sick. They want to see how the new technology and advanced therapeutics, how it works and how it help them. So they are more receptive to this type of advanced therapeutics. In terms of policy and things like that, of course, I mentioned that we work with TGA. They are so supportive of this kind of stuff that it's really nice to work with them, trying to identify a framework that they can just, say, okay, now you can actually provide phage therapy as a clinical service. And we've done quite a few health economic studies to actually demonstrate to the government that if we just implement phage therapy into the clinical service at public hospital, it would save the hospital itself a lot of money. So, for example, shorten hospital stays, improve quality, quality of care, but also, of course, seeing the outcome. So, for example, I can give you an example. We prevented a 7-year-old girl from amputation. So can you imagine, we saved the government NIDAS, for example, and we've actually shortened the hospital stay. So, for example, we treated a kid that was in ICU for over 400 days, and he was able to walk out of the hospital after a couple of months of phage therapy. And so that it's kind of stir out emotional response and go, oh, wow, this is amazing. But at the same time, you're a bean counter, right? You look at it and go, yeah, you've prevented and you've saved the hospital a lot of money.
How expensive are these treatments compared to antibiotics?
I think once we hone in on the biomanufacturing issues, we are able to produce an economy of scale of phage batches. They are very similar to antibiotics.
Fantastic. That's very good. So for
Hopefully.
so for doctors and members of the public, who basically anyone who's interested, who wants to learn more, Phage Australia, we'll put a link in our show notes to that, and people can follow your educational pages there and your potential. I think you do videos as well on that website as well about
Yes, yeah.
Phage, so that'll be a very valuable resource. So if doctors had patients who had problems with multi-resistance and curable infections, how do they get their patients to places like yourself? Is it only at Westmead Hospital for the moment, or are there other places they can be sending their patients?
So we operate under the STEM protocol. So we've got approval, and with that protocol, to access phage therapy, we need referrals. So for example, if you refer a patient, then it needs to be seen by 2 infectious diseases clinicians. So specifically infectious diseases, and both of them agree that this patient need phage therapy, and then we would actually take on the case. So then, we will organize looking at the patient isolate and looking at matching specific phage, and then go for production, or if we don't have it in our biobank, and then go from there. So I think people might think, oh, but I just want to have access to it. It's at the moment we try to put a framework on it so that we don't just get random cases. We actually do need to actually spend a lot of time looking at matching the exact phage to that
So the
patient's
pathway has
infection.
to be through a state government infectious diseases service, and then there'll be a tertiary referral if that's what's required. And look, things have to start this way naturally, and the technology becomes more robust and more replicable. Then, of course, we'll see the access broadened and made much simpler. Hopefully, one
Yeah,
day
and
we'll see it available in the community through primary care. But I guess that's still a few years away, at least from what you're saying, as we build up the technology and the commercial production of these phages. But it's very
yeah,
exciting, very, very
it
exciting.
is. Hopefully, so you mentioned it's not just Westmead. So with that protocol, we actually have over 25 hospitals that have signed up and have permission, site-specific approval to implement phage therapy at their jurisdiction. So we've got hospitals in Wagga, Canberra, in Adelaide, in Queensland, and also in WA that actually have implemented this protocol and treated patients with the phage that we produce.
Well, Professor Lin, this has been a very interesting discussion, and I think the audience has learnt a lot from this. And hopefully, if we keep our eye on your website and follow the developments in phage therapy, we'll definitely be able to help patients with serious infections a lot more. And I really thank you for your time.
Thanks for inviting me. We haven't even scratched the surface of it, but I'm glad, I'm able to have the opportunity to talk about the work that we're doing.
Yes, no, that's fine. Of course, there may be opportunities in the future to go deeper into certain aspects of it, but I think it was important for people to get an awareness of what's going on, because I think for many, even for many clinicians, they're still not aware of what's going on, and certainly a lot of the public's not aware. So this has been a good entree, let's say, and maybe we can touch base in 6, 12 months' time and see how your project's going and see how things are developing.
Yeah, nice.
So
Thanks,
thank you very
thanks.
much. Thank you.